This is the main building of our esteemed university, Copenhagen University. It's 850 years old, so it has a lot of tradition behind it. This is not the actual buildings that we are in, because the university is spread across Copenhagen city. In 2022, as you heard, I received the Nobel Prize. And when you receive a Nobel Prize, first of all, you immediately become like 20% more intelligent. And the second thing is that you are allowed to talk about things that has nothing to do with your chemistry. I shared the prize with Barry Sharpless and Carolyn Bertozzi, as you heard, and these are very good old friends of mine. I've known Carolyn since she was a PhD student. And I think they are both fantastic people, and I'm very proud to share the prize with them. When you get the Nobel Prize, as I said, you're also allowed to have an opinion, and my opinion is that chemistry is everything. So there is nothing around us that is not atoms or molecules that interact with each other, and for this reason, I think it's extremely important. I mean, it's chemistry when we are hungry, it's chemistry when we are sick, it's chemistry when we fall in love, it's chemistry when we make new materials and so on. So the young people should learn chemistry just like they learn to calculate and learn to write and read. They should learn chemistry from first grade, just in a way that we are meeting their demands rather than they meeting our demands for formalism and so on. I'll come back to that later. Another thing is research is a curious thing. So research cannot be performed with restraints. You cannot decide what to make for 5 years and then go ahead and do it. Because a lot of research, a lot of the groundbreaking research is actually serendipitous. And this means that you should do the research that you find is most important at every single moment. And therefore, funding by performance would be much better than applications. That means that we as researchers should allow an evaluation of the last 3-4 years. If we are very active, we get more money. If we're less reactive, we get less money or no. So chemistry is everything. That means that chemistry is also the key to life. So there are 5 processes that characterize everything that goes on around us. It's actual components of life. Molecular recognition is probably the most important one. So molecules, I come back to chemistry of life a little later, but molecular recognition is important for everything that goes on in biology. There's not a single event in biology that is not preceded by molecular recognition. And molecular transformation, that is control of reaction from one compound to another. Molecular signaling, so from the outside of a cell to the inside, for example, and transport across membranes and across organs inside the cell. And then molecular replication, of course, which is the basis for evolution. So this is the 4 components that makes up everything, 5 components that makes up everything. And chemistry is not good and it's not bad. Chemistry is just the knowledge that describes everything around us. And the atoms of life is actually quite few. If you look at the elemental table, these are 16 atoms that are the main atoms that compose life. So life has selected these only 16 atoms, and they make such a complex world out of those atoms. And that is because of inherent properties of the atoms and the molecules they can make. So atoms have, of course, a nucleus of protons and neutrons, and they are surrounded by clouds of electrons that are captured by the positive charge in the nuclei. So the electron clouds are shared between nuclei and the molecules are formed, and this means that there's a lower energy if we make a bond. Molecules, they can organize in nanoscopic structures. So we have both the formation of polymers, of course, but we also have these polymers coming together and forming nanoscopic structures with nano interactions. Carbon is central. It's central to the periodic table, and it's also central electronically, and therefore it can bind strongly to itself. So that is the basis of life, that carbon atoms forms very stable bonds with itself. Functionality is created when carbon bind to other atoms in polar bonds. And carbon makes double bonds, which is a prerequisite for life. That means that silicon life is impossible because silicon don't make double bonds. So the molecular properties, they come from the environmental response. So there's a lot of difference to a molecule, whether it's dissolved in chloroform or water. You can see that in nuclear magnetic resonance spectrum. The electron density around the nuclei is completely different depending on the environment. That means that every single atom actually in the universe is a little bit different from all the other ones. And that's what leads us to the complexity of life. And it's actually not possible for life not to occur because this is an inherent property of the atoms. So life will occur when the conditions for life is met. So there will probably be life on many, many planets around in this vast universe. So where does my interest for chemistry come from? Well, I was a young kid here in my grandfather's farm, very privileged. I could play in this beautiful place from morning to night, and I did so. I was interacting with nature every day, and I was starting wondering, what is it that makes this amazing complexity, this amazing synergy between different living organisms, including the bugs and the plants and everything. I didn't have the answer at that point, but later on I, of course, discovered that this is all due to chemistry. So this was my first inspiration. And this also led me to think about what is it in me that is really important. Of course, factual knowledge about what is going on between all these organisms is important. But it's also very important to have intuition and imagination, being able to, imagine this forest once you come home and you are lying in your bed and dreaming about it, to be able to associate the experience you have with other knowledge that you have about everything. Also, the ability to observe is extremely important, and to combine your observations and your associations and your imagination. This opens up the possibility of being creative and making something entirely new, and also have the courage to do so. And nowadays, we can actually use computers and computation to actually enhance our creativity. So it's not taking away anything from us. It's just adding. It's a tool that we can add to our toolbox to help us being very creative. And then perseverance, and I'll come back to why that is important in a while. So these is actually the properties that I'm looking for, the capacities I'm looking for when I have to employ a new candidate here. So the forest is fine, and I got some inspiration there, but the real inspiration came at 15 years old. I took a trip down to a company south of Copenhagen, and I bought everything they had which was reducing or oxidizing, and I made entirely new powder formulas to make fireworks that was just spectacular. And this is not my firework, but it looks pretty much similar actually. But as I said, the perseverance is important, and that's because the road to scientific success is never straight. It never goes like we want it to. And that is because nature is very complex. So science projects made at the office desk almost always fails initially, even when it's your very clever supervisor who have made them. So you cannot plan, and you really have to look in every corner of your experiments to look for opportunity. So you will fail, but you will rise again. You'll use all the infrastructure and instrumentation to analyze what happened, and you will find out better ways to do things. At the same time, time is precious, so you should really use it optimally. And also, you should do what you find is interesting. You should get out of your comfort zone and do what you think is interesting. So you have a great advantage if you change field occasionally in your career, change it to peripheral fields which can bring in new, you can bring in new knowledge to this field from your previous field, and you can also take advantage of this new knowledge that you get, which means that you have to study all your life and also bring your experience to use in the community. So this is T-Rex. Let's call him Ronald T-Rex. And as you can see, he is not burdened by social skills or empathy. Rather, he is controlled by aggression molecules like adrenaline and noradrenaline. And he is probably also caring a little bit for his kids. But his brain is very small. It's a huge animal with a small brain. And besides these two molecules, he has a simple pleasure molecule here, dopamine, which satisfies him when he gets something to eat, or serotonin, which works in his cognitive operations. We have also this simple dinosaur brain, but on top of that, we have a layer of brain that is aiming at controlling the very simple functions of the primal brain. So medulla and amygdala is in control, but we try to control them by counteracting from the large human brain so that we can function as social individuals in a society. If we look at the dopamine, it's a reward function. So when we get dopamine in the frontal cortex, this is a nice feeling, which stimulate us to do the same thing again. So when we have a nice situation, we will try to redo that. And the serotonin pathway controls a lot of the motor function and memory, processing, sleep, cognition. And that's also a very important thing that we should not mess up too much. So here is a more complex molecule. This is oxytocin, the love molecule. And this is a unique molecule which is excreted both in women and in men, but it's also the lactating molecule that allows women to generate milk when they have a newborn. The structure is shown here in 3 dimensions. But this is not what it actually looks like. What it actually looks like is this, like a chewing gum. And you can see that this is the electron clouds that is deficient or have a surplus of electron density in different areas. And they try to interact with the opposite in a receptor that binds to this and triggers a reaction inside the cell. So you can also see here that nature is very cheap, because here's another molecule, and this molecule controls the blood pressure and also the heartbeat. And this is vasopressin with the arginine instead of leucine and a phenylalanine instead of isoleucine. So nature is cheap. It reuses the molecules again and again for different purposes. But you can also see that it relates the heart functions here. So if the oxytocin binds to the receptor, there's a signal sent to the interior of the cell where this huge complex allow a number of different functions inside the cell. And that then leads to empathy, love feelings, pleasure, nesting functions, and much more. So there are many different receptors to these old molecules that has been there since the beginning of humanity. Peptides. Peptides are really important in nature. They are nature's toolbox for signaling, for functionalization, and so on. And it's a boiling pot of research opportunities because we have peptides as materials, as drugs, nanostructures and nanoparticles. Virtual peptide design is possible, so we can use computers to design our molecules. Peptide biology and biological chemistry is a very hot field that takes in a lot of peptides. Peptide can be used in combinatorial chemistry because of the huge diversity of peptides. In particular, if it's chemistry, we can take advantage of about 5,000 different amino acid derivatives. It has also promoted polymer chemistry because a lot of peptide chemistry is associated with polymer. And de novo protein design is now possible today. We just had a Nobel Prize for AlphaFold and for the AI that can look at matching molecules. Peptides can be catalysts. We can make organozymes or microproteins with function. And then we can combine organic chemistry and peptide diversity. So this is a really, really strong field. And I started out in Carlsberg Laboratory trying to exploit this functionality. I started at DTU as an engineer, the Technical University of Denmark. Then I went to Cambridge and came back to University of Copenhagen, was there a couple of years, and then I came to Carlsberg Laboratory, which is this famous building. There's a huge part behind it as well, but this is the old building where the chemistry department was. I built a lot of laboratories, and then I built a technology platform. And much of our chemistry really is based on the PEGA resins, so polymers which we synthesize ourselves, which are polyethylene glycol based, so the structure of it here. And we were able to do a very large number of organic chemistry reactions quantitatively on these supports. We could combine these reactions with peptide chemistry, and we could investigate enzymes, different proteases, transferases, and even whole human yeast can be mobilized inside these polymers and used. So I got this idea that we should have quantitative chemical reactions for CompiChem, combining quantitative chemical transformations with peptide diversity, and then use that for drug development and adding to STEM in society in general. And this was the foundation of the large grant I got from Danish National Research Foundation. So there we built a platform based on combinatorial chemistry, so we can synthesize millions of compounds, and we can screen these. We typically synthesize 100,000 compounds, and we can screen these with enzymes. Here's 6 active beads in an enzyme assay. We analyze what was active and what was inactive. We have sorting equipment. We have mouse experiments here, where we take one of these enzymes and inject into the mouse, and we can see that the enzyme is active during an arthritis, induced arthritis here in the foot of the mouse. We also make catalysts, palladium catalysts, for example. We make a lot of pharmaceutically interesting compounds by a new click reaction that I will not talk about today. We can do cellular studies on these polymers because they are biocompatible, and with this peptide here, which is derived combinatorially, we can harmlessly make a monolayer of cells on the surface of the beads. They actually behave very well. And here you can see an assay in which we have the adhesion molecules, and we have the cells adhesion. And then we also have a assay in the cell where we get fluorescence upon receptor interaction. So this bead here in the corner is an active bead, and these are inactive beads. So we analyze what was on there, and then we can resynthesize and make more advanced studies with that. We also have a microparticle matrix encoding, so an optical encoding of beads shown for a single bead here in the corner. We have a machine where we can read the structure of the bead. Every time we do a reaction, we can read this microparticle structure or matrix, and that allows us, after the synthesis, to decode the structure on the single beads. The way we make the polymers so accurately is by having a mixing of macromonomer and initiator, and then we form droplets that are passed through a heating coil and then collected on a filter. And we can do this in a very small suspension medium, a very small amount of suspension medium silicon oil that is transferred with a gear pump in this recirculation. So we can make the beads with very great precision, as can seen here. And the best one, which is the one in the picture here, is 415 on average in microns. And then we have plus minus 9.2 micrometer. So we can even do quantitative assays on these beads. During this period, we worked with acetoacid chlorides in order to make very difficult peptides like the one shown here. We thought that this is a good reaction, so maybe we can even isolate a carbon nucleophile like the copper acetylene here. So Christian Tornø, who was a PhD student at the time in my group, he made the copper acetylide from the alkyne, and then he coupled it with the acetoacid chloride, and that was actually quite difficult. It should have been very easy, but it proved to be quite difficult. So Christian worked several months to try to do this, and I was getting impatient. He came to my office and said, maybe I shouldn't work here, maybe I should rather work on Novo Nordisk. And I said, take it easy, let's look at it. And we did, and we found that actually you form exclusively this compound, even in the presence of this hyperactivated acid chloride. So we could use the acid chloride subsequently for further reactions. And that was quite surprising. So I asked Christian to investigate the compatibility with all the amino acids, and he did so. And here he is presenting the result at the peptide meeting in 2001, Wave of the Future at APS. He was very happy for the results. But what is this click chemistry reaction? So here we have an alkyne with this HOMO of electrons. This is one of the two pi clouds of the triple bond. And that is very high electron density here in the center. So it looks out for positively charged ions. If you have copper 1 in there, it binds extremely well to acetylides for some reason. That acidifies the proton. It sucks out electron density and acidifies the proton, comes off, and the copper takes the place. Nothing much happened with that, but a friend comes in here and attaches itself to the electron cloud, pull off the electrons, so it sort of takes off the electrons like when you open a can of sardines to get into the good stuff. And here, what we have here is actually the LUMO, or the empty balloons or orbitals, if you will, of the alkyne, with the two metal ions equally situated here at the end. And this looks out for electron density. So if we have an A side, it happens that the A side has a HOMO, or a full electron cloud, that exactly matches the electron cloud of the LUMO of the alkyne. And this is the reason for the very high selectivity of this reaction. So we get a ring closure first, we get a metallocycle, and this metallocycle loses first one copper atom, then it loses a second copper atom, and this is Christian today, and he is now leading the click chemistry department at Novo Nordisk. So it was a good thing that he stayed. There's only one caveat with with the click reaction, and that is the oxidation of copper 1 to copper 2. This happens when oxygen is present, and for the ideal click reaction, it wouldn't matter to have oxygen present. But here it does, and Sharpless solved this by adding a reducing agent that takes it back to copper 1. But what happens when you do that is that you have this redox cycle racing around many, many times. Every time it goes one round here, it generates a lot of reactive oxygen species, and that destroys biomolecules. So here we have a protein that was clicked in the presence of oxygen, and you can see plus 16, plus 32, plus 48. So these are all the oxidized proteins. And of course, we cannot revert that. So we are really at a loss if we do this in presence of air. So we have to remove oxygen, avoid redox cycles, and use protective catalysts that maintain the copper 1. Here we are. This is in Stockholm. This is one of the greatest vacations you can possibly go to. I think that we were invited to Stockholm, and I can really warmly recommend this holiday. Here we are oriented about the week, what is going to happen, and you can see here for the greatest benefit to humankind of Alfred Nobel. And this prize is still extremely important, of course. So when you're up there, you are delivering something that meant a lot to your research. And this here is the first reactor we used for combinatorial chemistry. It has 20 columns, and I made this in the garage of my neighbor. He had a lathe, so I actually made this myself, and it's now up in Stockholm. This here is a dress, one of the 6 dresses for the Nobel Prizes that was created by the Department of Arts in Stockholm University. And there were 6 other dresses, but this one is the important one because this is the Click Chemistry dress. Here you also sign a chair, and you can see Manuel Carpentier here, so that's a very prominent position that my signature is on. Now, what can we do with click chemistry other than just connecting phenyl group to a metal group? We can do a lot of different things. So we can express proteins using amper stop codon technology, which allows to include in the protein these click residues. We can also synthesize compounds like this one with a site here. And this could be a recognition molecule, for example, an antibody or something like that, with 2 alkynes. And if we mix these 3 together, we'll get what we could call a molecular robot, which have the properties of the 3 constituents in one molecule. So this molecule can find something, it can penetrate, and it can kill with this guy here. So here you have a cancer cell. And that is what is made feasible by the click chemistry. Now the ninja takes the other molecules into the cell, and the cell is dead by programmed cell death. So we can make robot molecules, but as I will show you, we can do ligation, peptide protein stapling, surface immobilization, polymer synthesis, CCPs and ligands, affinity and activity profiling, and not unimportantly, we can do green drug synthesis because all of this click chemistry can be carried out in water at the last point of construction. So just to give you one example from the 3 I had in the previous slides, this is the cell-penetrating molecule that we made. It has a cargo, it has a hydrophobic region, and it has the charges. And we made actually a large variety of different molecules with the click bridge here and here with different fatty chains here. And we found that, so we had these 4 building blocks with O or N, and these were synthesized by Ian Zhang in our laboratory. And we found that this one combined with that one is the only one that is really efficient out of 16 molecules. So you can see here our CPP15, which we call it. It enters into the cell, and this is a merged image from the cell surface and the import. We tried a lot of different combinations, and this is the TAT peptide, which is a very good CPP. And here we have our molecule, and you can see it's even better than the TAT by quite a lot, actually. And when we do the assay over many different cell lines, we could see that it was almost uniformly active across many different cell lines. We could also demonstrate that it was dinosaur inhibited. That means that it was a dynamine promoted endocytosis that we were looking at. And this is what the molecule looked like with the fatty chain sticking out here and the 4 positive charges surrounding it. And we connected this to a GFP. So this is a molecule connected to the GFP. And here you can see the entry into cells of the GFP modified peptide. And if we do the GFP alone, like here, we get nothing in. If we mix the GFP with the CPP, we get nothing in. And when we have this connection, you can see how well the molecule enters into the cells in the endosomes. We have great hope for this as a blood-brain barrier transfer reagent as well. So we can do click ligation because here we had a click function to connect to the CPP. And another example of how we can use the click chemistry is here for stapling. These guys are 400 million years old, not those, but the species. And they behave like a pharmaceutical factory. They make a fantastic turquoise blood that we harvest, and we use 2 macromolecules from the blood to measure bacteria in blood, in our own blood in hospitals. But it also have other very important antibiotics in the innate immune system that it possesses. And target placing 1 is one of those that can kill bacteria, shown here in the corner. And it looks like that. It's a small peptide, a beta hairpin with 2 disulfide bridges. We were considering, can we actually replace the disulfide and get a bioactive compound if we do the click reactions instead of the disulfide bonds? In this process, we also found by NOE experiments, so NMR, nuclear magnetic resonance, we could see the interaction between 2 of these molecules, meaning that they actually form a dimer and hide away, under normal circumstances, all this inner part, which is used for the formation of the pores of this molecule, because that's how it functions. So this here is what it looks like. It's a benign with positive charges swimming around in the serum. And when it meets a negatively charged cell membrane, it aggregates on the surface and it forms forms the monomer, and then it makes a hole in the cell, so a pore, and all the interior of the cell can then escape from that pore. So does that work in reality? We tried 4 different bacterial species. So this is coli, staphylococcus, epidermis, Salmonella typhimurium, and Bacillus subtilis. And we asked if this compound would kill our compounds, or the natural compound would kill these cells. So the smaller these small cones are, the more active the compound is. And the first row here is the target placing itself, so the natural compound. And you can see our compounds is almost as active as a natural one, even in the cell where, so this is Bacillus subtilis, where this target placing is completely inactive. So it could be that Click is a good mimic for SS bonds that can improve macrocyclic antibacterial peptides to avoid resistance. And the resistance is probably due to disulfide bond reduction. This is CuAAC Click mimicry, and we would like to also show other applications. But for now, we are back in Stockholm. This is the great day where you give your talk. You are very nervous because like now, I'm talking to a lot of people in India, but here you're talking to probably 10 million people. And it takes a little bit before you can reach your poster and deliver your talk. But it went well, and it went well for all three of us. And here you can see we are very happy that it went so well. So we work, as I said, with molecular recognition. And we thought that antibodies that are the penultimate or the ultimate recognition molecule in nature, they are very big molecules and almost impossible to synthesize. We are a synthetic lab. Maybe we can do something else. This here is a beta hairpin. It has a very well-defined structure because of the threonines, which gives extended chains, and because of this click bridge at the end of the molecule, together with the turn in the other end. That allows us to actually combinatorially change all these red residues. So we can fit this molecule to any kind of protein surface. And this is an example where we have taken interleukin-6, an immune molecule, and we made two of those. First, we designed one up here, which is attached to a bead after synthesis. We have made another one by design down here. I'll come to the design process soon. But we made another one here, which interact on the other side of the molecule with a fluorophore. When we mix the bead with this molecule, we get nothing. And then we add 20 nanomolar interleukin-6, and you can see what happens. The peptide on the bead recruits the interleukin-6, and the bound interleukin-6 recruits the other molecule from the solution. So this actually is a very good proof that this design process works very well, because we have made two designs in one go, and both of them worked. So can we use this kind of design for anything? We thought that Alzheimer is a really big problem. 65 million people has Alzheimer in the world, and that's quite a lot. And it's very, very expensive for the community to actually take good care of so many people. So this is an Alzheimer plaque engrammar at Allied isolated these fibrils from the actual plaque and did an electron microscope structure. And this is a structure. And if we now divide this structure in two here in the middle, we get the growth surface of the fibril. There's one growth surface here and one on the other side of the molecule, which we can then design these kind of beta bodies or beta hairpins to, and then create molecules with high affinity. So we did that, synthesized two beta hairpins that can connect to either end of the fibril to prevent aggregation. And you can see we have a little bit of fibrillation. This is a fibrillation assay in which this fibril binds to this fluorophore and give us this signal. So a little bit of fibrillation initially, and then it dies out. If we have no compound added, we get very good fibrillation curve here. And then when we add the peptide to the already formed fibrils, we have a great reduction in fluorescence associated with defibrillation. But we were not quite happy. So Yuan Zhang, PhD student in my lab, he synthesized a compound where we have protected glucosaminic acid on the nitrogen with FMOC. Then we have treated that with triethyl silyl chloride to get the TES derivative. We hydrolyze the carboxylic ester here, and we get a building block that can be incorporated into peptide synthesis. And we did that. It has the same configuration as D-threonine, so these are all D-amino acid hairpins, which is, of course, an advantage because they are not metabolically processed. And also, we found that they are completely non-immunogenic. So here we see 3 of these peptides with the glucosaminic acids in various places. And these are the 2 most important ones, number 3 and number 5 here, which binds strongly to the 2 growth phases of the fibril. And when we mix these compounds with a beta 1 to 42 or beta 1 to 40, the peptides from Alzheimer, you can see a complete inhibition of fibrillation here, both for the 1 to 42 and for the 1 to 40. So that was extremely satisfying. And what we think happens is that you get this kind of small fragment of the fibril that is terminated by the 2 peptides. And these 2 peptides carry a huge coat of water with them, so they help for the solubilization of the fragments. And then during our deep sleep, we can most likely wash some of this out every night. Martin Wolfram was another PhD student in my laboratory, and he was interested in seeing if we could actually cleave the Alzheimer peptide. So this is a nucleation site of Alzheimer peptide with the 2 phenylalanines sitting here. And we made a FRET substrate, so the fluorescence quench substrate with a nitrotyrosine in one end and an aminobenzoic acid in the other end. So when we shine light on this one, it's dark because the fluorescence is tunneled, or the energy is tunneled here to the nitrotyrosine, and we see no fluorescence. But if we cleave it, it will fluoresce. He made carbene peptides like these ones, where a metal ion is bound to a carbene, which have ability to bind to this substrate. We also use a bipyridyl residue here for this purpose, again, to bind to the substrate and see cleavage from the metal ion. Martin was very happy because it worked extremely well. So he sort of reduced the amount of catalyst to half, and it still worked very well. So he removed it again completely, and it still worked very well. So that was very strange. That is when you have to use your observation and think maybe something strange is going on that we should investigate. So what happens is that we have an autocatalytic process of the Alzheimer peptides. They aggregate and form proteolytically active aggregates. This could be a biological intended function, or it could be just by serendipity that these activities are observed, but the activity is highly specific to this central region, which is exactly in the nucleation site of the fibril formation. So we looked carefully on this by synthesizing all the different fragments you can think of from the Alzheimer peptide, and we then investigated it by LCMS. So we start out with the red compound here, it's there, then we form F0 after 11 days, and then here after 12, 13 days, and so on, and then we eventually get to complete degradation in this region here. So these are the fragments that we can isolate during this cleavage reaction, and that proves the cleavage. We can even take a small amount of the very active intermediate here and add it to a longer peptide, like 1 to 28, and after a short initiation period, so this is just 1 microliter added to a larger portion of the peptide, and you can see after a period it starts to hydrolyze. We can even do so for the very long peptides. Here you can see after 12 days, without addition of anything from the active components, we still have the intact peptide. But if we add the peptide from the active components from up here, we see completely cleaved products. So there seems, if we leave this for a whole year, we start seeing cleavage products for those as well. So even the full-length beta can do this with time. This is an important observation. And we also saw something else in this investigation. We added this to the mica chips and ran atom force microscopy. And what we see here is that initially we form this single chain donut. So that's the start of the aggregation. So you have all the peptides lined up together here to form a donut. And that donut then collapses. We have many examples here in these slides of collapsing donuts. And they form these small pins. And they then find each other terminally and react slowly to form eventually a more mature fibril. So this is sort of the entire mechanism of fibril formation that is elucidated here. So we have the mechanism, we have these donuts, and this is a 3D AFM of the donuts. And we have this theory that we form an aggregate of many different peptides, different lengths from the Alzheimer, that has enzyme activity, acts like a protease. Interestingly, actually, if I just go up again. Interestingly, there's a paper from 2015 which describes the death of neuronal cells in the area of plaques and correlate this with the hydrolysis of NCAM molecules, or the surface adhesion molecules of the neuronal cells. And when we look at the sequence that is cleaved, it's almost it's property-wise, it's almost identical to the cleavage site for the Alzheimer peptide. So that is very interesting in my opinion. Here we are back in Stockholm, and this is the day where you have the ceremony. You meet the king of Sweden, and he hands you over this the thing here, the medal, and you get the medal, and he says, "Thank you very much." And then the king actually takes it back again, and he goes and sits down, and you wonder what happened there. But the reason for that is because a lot of us made the inventions like 20, 30 years ago. And since then, we maybe even have a little bit of Alzheimer ourselves. So a lot of the Nobel laureates have forgotten their medal in the ceremonial hall. And nowadays, you get it at the last day, you actually get the medal, and then it's your responsibility to bring it back home. This here is an HIV invasion of a CD4 cell. So what can we do? Can we have an intervention that prevents that? So what happens here is that you have the GP41 and GP120 here at the surface. The CD4 receptor binds here. You have a co-receptor. You have a fusion peptide that goes into the cell membrane from the HIV particle. And then it forms this hexahelix. So there's like two helices here that can come together and form 3 helices here and here that come together and form a hexahelix. That leads to a hole through the membrane, and that is how it injects the material from the virus. So what can we do? This is a crystal structure of the hexahelix. So we thought maybe we can just take this hexahelix and make a really good affinity probe for the triple helix that is in the center. So we made first the triple helix in the center by click chemistry. So you can see we have made 3 triazoles here, And this is just showing the template with the fluorophore, lysine, and 3 A-side groups that it's coupled to alkynes on the peptide. And these are the 3 peptides attached to this template. So that looks like this. And now the other 3 helices is bound like that. So we just take one of the helices off, and we make a beta body that fits extremely well to the surface. This is the product, and you can see we have now made the beta bodies here on the resin. And now we can come with a triple helix with a fluorophore, and it will find the bead and bind to it, and we get a fluorescent bead like this. So that worked extremely well. You can see here our beta body. This is actually the natural helix that binds to the triple helix. And we see that the interaction here is much weaker than our beta body interaction. This is a bead without anything on it in the same conditions. So this is the bead without anything on it, and we add this peptide here. The fluorophore, or this peptide with the fluorophore, is quite insoluble, so we actually had to add some urea to this assay in order to make it work. But we can do CuAAC click molecular assembly of 3 helices on a scaffold, for example. And we can also do surface modification. So what we have here is a ceramic chip with hydroxyl groups on the surface after oxidation. We can transform those by aptase into amino groups, or aptase A side into aceto groups. And then we can either use chemical couplings to the amines or CuAAC coupling to the A sides. And this is what we show in the next couple of slides. So here we have used that one, and we can chemically modify nanowires, for example. These are nanowires that are only 80 nanometers across, and you can see how nicely we can modify those, both with the chemical modification, but also with click chemistry. So we have made this click substrate and immobilized these here on the nanowires. And this is a fluorescence quench substrate, so we will see a red color when we cleave this with, for example, chymotrypsin here. And that is giving us red colors on the pins. It's not extremely visible, but you can see the cleavage compared to the dark image over here. We can also immobilize things with here we have used actually Caroline-Bertozzi's click chemistry. So first we immobilize the click component using conventional chemistry to the amines, and then we can come in with a A-site modified GFP, and that can then react with the alkyne of Caroline's chemistry. And here you see the modification with GFP on these nanowires. In order to demonstrate that is actually what's happening, we also had an antibody that recognized GFP, and that is bound to the same chip here. So you can see it's really GFP that has been immobilized. So click materials, functional surfaces can be made with click chemistry. We have here now described a couple of click reactions, the spark reaction and the CuAAC reaction. And then there are 2 other reactions that I consider click chemistry. One is a TCO reaction, a fantastic reaction, which is a Diels-Alder reaction with loss of nitrogen. That gives us this functionality, quite large compared to the click functionality, but it's very versatile. And then we have, and I've not described this today, but we think that we have a very good click reaction here, which is intramolecular and driven by delta S rather than delta H. So this cannot be used to link molecules together. It can be used to make stapling within molecules. but it's completely selective. It forms only one stereoisomer, and it's completely radius selective as well. So we have 4 click reactions, 2 of them driven by delta H. The TCO, where the equilibrium is shifted by loss of nitrogen, and the enaic reaction, which is delta S driven, based on the Gibbs free energy relation. We are back in Stockholm. This is a great day. This is the day of the dinner. And here you see my wife, and she is extremely proactive. So when she saw the dress on the first day on the mannequin, she called up the arts department of University of Stockholm, and said, "Well, I need this dress for the dinner. Can you do that for me?" And they said yes, because that was a win-win situation. So they very quickly actually made a whole new dress that fits her. And we went to the dinner very happy together. But when you dress your wife in this fantastic fashion, you can actually see here that this is really artists that made this dress, because we only got 6-membered rings, no 5-membered rings in this dress. But when you dress your wife like this, there's a great risk. So this here is Prince Philip of Sweden. He spotted my wife and very quickly separated us. So I'm there behind somewhere. And she was seated between Prince Daniel and Prince Philip of Sweden. And I was somewhere else in the room. So that was a disappointment, because I wanted to sit by my wife. But it doesn't really matter, because the next day we was at an intimate dinner at the Royal Castle in Stockholm, and I got the sweet spot. This is Victoria, the coming Queen of Sweden, and she is an amazing person. So I was very happy, as you can see. So back to this thing about use and education. I think the young people are completely different today than they were when I was young, and they need to have entertainment in order to learn. So my philosophy is that we should make actually small videos, like 5 minutes for the first year every week, 10 minutes for the second year, 15 minutes for the third year, to explain in a consistent way how chemistry works. So we can, of course, use material from the internet as well, if we filter it and index it and license it. But it's not consistent, and the best way would be to take a completely consistent new version of chemistry, put it into videos, and let our young kids have it. So we call this AI, and it's very helpful. So you just tell which grade, which week, and it's delivered. You get a video, VR or whatever, it's produced, and then that can be both a helpful hand to the teachers and also a great way to teach the kids in a way that they actually experience something. So today it's thought that kids only want to do experiments because it's fun, they want to play. But I think that they really also need to understand why. And this is why videos would be very good, in particular if we could get some personality to the atoms and molecules. And this, whatever language you make it in initially, can easily be translated and globalized. So I think that would be a very good idea. Also, I sent this small invention, I would say, to Lego in Denmark, and asked them to make a molecular building set that would actually behave like real molecules behave, using magnets for hydrogen bonds, using shape for introducing barriers for rotation around the bonds, etc. So when they assemble these molecules, they will actually behave like the real molecules. I think that would be very, very useful. So the teacher draw a drawing on the blackboard, and the kids can make it. So understanding of chemistry is more needed than ever before. The good life is a simple life. All we need is food and water, social acceptance, reprocreation, roof, clothes, should be clothes, hygiene, should be hygiene, health and survival, freedom. These are the basic things that we need for life. A lot of people don't really understand this, but I think a chemical education would amplify the understanding of what is it that makes a good life. And the positive consequences of early global introduction to chemistry could be monumental for our environment. We could get more people involved in the problem, the challenges that we are facing at the moment everywhere in the world with global warming. So for the climate and environment, for global politics, so people would actually elect those which understand the chemistry and the consequences of what we do. For conflicts, so understanding chemistry allows us to set aside conflicts. For more equality, understanding what the simple life is. And for resources, so we would know that there's an end to resources. We would know that we have to make best use of them. For wealth distribution and for peace eventually. So I think these are very, very important things to teach the kids already now. So what is it that shapes the talents? There's many factors that influence the choice of education. So knowledge, but also the parental influence. So the parents have to actually teach the kids to make the right choices. Institutional influence, the authorities in the school, their own courage, so that could be trained as well, courage to actually make the right choices. Status and opportunity, so sort of a more egoistic perspective on what can I do with my education. The salary, what do we pay people for their services? And then I think the most important one for me is the existential curiosity about the universe, about the life on Earth. Life is changing the surface of Earth at an unprecedented rate at the moment. Or if we set aside the extinction events of meteors and so on, life has never changed the surface of the Earth as fast as we do right now. Education relate to identity, and identity building is happening extremely early. So already in the first grade, there's a lot of identity building starting, and it's largely emotional in the beginning. So it's very important that we address the emotional side of learning. And we should be careful where they get the influence from. They can get it from parents, from friends, from authorities, from media. And the media is, of course, a problem at the moment. But then they also can get identity by knowledge, and this is why we should let them have the knowledge about our environment. Because we want the education to match the societal needs. So how can we optimize this selection? And that is from this identity building. Efficient education should happen without pressure. Because we cannot learn if we are forced to learn. We can only learn when we want to learn. And that means that we have knowledge versus authority here. And positive reinforcement as self-esteem is extremely important for taking the next step in your life. So we start early and reward selection when it's done right. And excellence, once it is created, is not limited to a single one who is excelling. The excellence is actually when you can make a playful team with a well-equipped playground, meaning that we will have in science all the infrastructure that's required to do the job we need to do. Excellent is freedom of thought, not for the PI, not for the leader of the research group, but for everyone in the group. And it's driven by curiosity, so you should spur the curiosity of all your co-workers. Excellent is true engagement. You have to dream of it before you fall asleep. If you don't, you're doing something wrong. And then you do it if you are a student and your PI is there, he should allow you to do it. And that's freedom. So you get independent co-workers with their own ideas. That can be difficult, but it's much better than not having it. A sense of self-determination is generated, and this is happening under responsibility to actually carry out stuff that is needed. So you challenge the skills and move forward while you are modest yourself as an excellent person. And most importantly, excellence is not something that is created behind an office desk. It's by practicing the chemistry and observing. And all of the above is done while keeping track of the greater picture of the research programs. So then the last slide here, facilitation is true funding without constraints. Because most discoveries, including all the Nobel Prize discoveries, are disruptive discoveries that are serendipitous, and then they are leading to things which you couldn't imagine later on. We need productivity, quality, and creativity and responsibility, but we should not do this by force. So today, most countries have all their research money in one big pool, or several big pools, but put together, funding either applied or fundamental research. But these should actually be separated in the society. So you have a pool for fundamental research funding and another pool for applied research funding. And you should decide how much research do we need to be competitive tomorrow, and how much do we need to make the bread and butter today. So with that, I would like to thank you all for your being here. I'd like to thank Danish National Research Foundation, Carlsberg Laboratory, and University of Copenhagen. My supervisor in my PhD study and later professor at Carlsberg Laboratory, Claus Bock, Michael Bolz, the institute leader who invited me to Copenhagen University, and Carlsberg Workshop, who did a lot of the actual instrumentation that I've used in these studies. I also would like to thank my co-workers, quite a lot of them. And I would like to thank the people at Carlsberg Laboratory over the years, contributing to all of this. And importantly, also the people from Copenhagen University, who's currently working at the further development of these chemistries. And finally, my family, Phaedra, Marie Saint-Hilaire, who's also a scientist, Ajani, who's studying chemistry, Anna and Sandra, my family, thank you very much for your patience over the many years.
Yeah, thank you. Thank you, Professor Meldal, for your brilliant and inspiring lecture. I want to express my deep gratitude to you for this really insightful presentation. Your childhood curiosity, creativity, and courage to explore has certainly led to an extraordinary journey of discovery. And now your concern for the youth and climate. You also emphasize the role of openness and serendipity for scientific discovery, as because scientific discoveries are mostly non-linear, as you very nicely told. So, sir, thank you, sir, once again. So I will be now asking some of the questions we received from our live participants. So also it is a general reminder for those who have not submitted their question. Please scan the QR code shown on the screen. And also the Google form link has been sent in the chat box. So please do submit the questions. Yeah. So Professor, we have got this question from Talpunur Rajsekhar Rao. He's in Education Department. And he's asking that what inspired you to explore the concept of click chemistry? And how did you how did your discoveries shape the way we understand molecular interactions in chemistry? So please.
Yeah. So the first question is what inspired us? What inspired us was the observation that we got a strange reaction that we had not expected. This is for me has been all the best things that we have done has been observations in the laboratory that something does not behave according to our expectations. And if that happens, then that is the inspiration to continue and investigate that in more detail and maybe discover something new because it's unexpected. So it's highly likely to be something that was not there already. So this is the click chemistry, and how did that then change our view of molecular interactions or recognition? I would say that even today, we do not understand molecular recognition. So a lot of people think that they do understand molecular recognition, but they don't because they cannot calculate it. So we can even go 10 million wrong in a calculation of molecular interactions in terms of the energy of interaction, which is, for me, a clear indication that we do not understand it. So that is by algorithm. But with AI, what happens is that we have 10 million or 100 million crystal structures, and the factors that we do not know, what we do not know about molecular interaction and which we cannot calculate, is actually captured by the AI because the AI combine all the information and have this information about molecular recognition implicitly given in the analysis of the data. So the answers we get from for example, AlphaFold are very good. And this is not because we can calculate it. This is by simply taking all the information of the database, crystal database, into a filter, which contains this filter contain the unknown information. So Click has not directly made a difference there, but Click has allowed us to combine several modalities, several a recognition molecule with an enzyme, with a penetrator, so you can make molecular robots that allow us to study biochemistry in ways we have never done before.
Thank you. Thank you very much for your thoughtful response, sir. So there is a second question. What is meant by nanoscopic structures? What is flow polymerization, and what are those bits actually? It is a question by Aritra Raha, Aizer Bhubaneswar.
Yeah, so the nanoscopics, that's two questions. So the first one is a nanoscopic structure. So the cell is an example of a nanoscopic structure. You have the cell membrane, which self-assemble, and you have the inner organs of the cells, which are self-assembly of molecules. You have just soap in your sink forms bubbles at the self-assembly of molecules. That is a nanoscopic structure. It's used quite a lot in nanochemistry, for example, where you are aligning things on a surface, proteins that are aligned on a surface, almost like a crystal structure in one layer. And you can look at self-assembly as the weak forces that are allowed by a specific molecular recognition between molecules. So also there, molecular recognition is the key to everything. And on the other question, which is about the polymers, these are polyethylene glycol, which is completely mixable with water and with toluene, for example. So it's both on the unpolar and the polar scale, very mixable. So we can actually make these polymers by adding a small amount of acrylates, which can be radically polymerized. And because we have a acrylate in both ends, we form a three-dimensional network of these polymers. So they are 90 to 95% PEG and a little bit of polyamide in the PEG chains. So this is very, it's like a sponge. So the polymer, when you put it in water, swells up 10, 15 times. And this is making pores in the polymer that are large enough that even large proteins can diffuse into the interior of the matrix, of the polymer matrix, and carry out reactions in there. So this is why we call it biocompatible polymers.
Thank you, Professor. Yeah, there is a third question by Pradyumna Sahu, IIT Hyderabad. Can we make a compound like triazole click type of complex so that they can attach to the cancer cell or diabetic place so that it's a game changer for human life?
Yeah, so this is all being investigated, for example, for GLP-1 and for insulin. It's been investigated now how you can use click chemistry to add things that modify the lifetime, the residence time, the activity, and so on of these compounds. Also in cancer, I know that the TCO click reaction is actually directly used as a cancer treatment reagent because you can connect something to this click moiety that will be released upon click reaction. Take the other click moiety, add that to an antibody that recognizes the cancer cell, and then when they click, you release the cytotoxic compound attached to the other part in the vicinity of the cancer cell and kill that cancer cell. So click chemistry can be used for that kind of purpose as well. And we are not finished with the project with the cancer treatment, but we are working on it. And we have the cell-penetrating peptide, and we have the recognition of an inhibitor called XEAP that's overexpressed in cancer cells. And we can definitely link these two together, and then we just need to have a selective delivery to the cancer cells. So we need a recognition molecule, and that's something we're working on. Was that the answers?
Yeah, I guess so. So we have a fourth question by Tuhin Sinha Roy from Ideal Engineering College, Kolkata. So he's asking, how do you envision the future of click chemistry evolving, and what potential application do you see emerging in the next decade?
Yeah, so I think that we should look for click reactions, particular click reactions which are orthogonal to each other. So we can do a CuAAC reaction in one end of the molecule and a TCO reaction in the other end, and a third click reaction and a fourth click reaction. All of those would be quantitative and give us very selective modification of a protein structure, a DNA structure, whatever we are working on, to make just spectacular molecular architectures. That's an art in itself, in my opinion. It's really artistic to look at the complexity that we can achieve today using synthesis.
Thank you, Professor. So there is...
And I'm sure there will be probably hundreds of those click reactions out there, but it's very difficult to find them because of all the properties that you need to install before you have a click reaction.
But we can use AI, as you told that we can use AI to search for that type of click reactions.
Maybe if we have but the AI has one problem. That is that the filter you install in AI is all the known stuff. And then you use the known stuff to predict very similar known stuff. Right? So if you use AI to predict a completely unknown protein like AlphaFold, it will come back with a little bit of fold, and then you will have a lot of doesn't know kind of thing. That's the same for chemical reactions. If you don't have anything in your filter, in your notes, that tells you what you're looking for, then you won't find it.
Okay. Yes, Professor. So we are continuously getting a lot of questions. So this is question by Dr. Bharat Pandey from Government PG College, Almora, Uttarakhand. So he's asking, if click chemistry were a molecular matching service, How does the CuAAC reaction ensure a perfect match between azides and alkynes? And what makes copper the ideal catalyst cupid in the world of drug design and bioorthogonal chemistry?
Yeah, so as I said in the lecture, copper has a very, very high affinity for alkynes. There's like hundreds of crystal structures of alkynes in complex with copper and in the Cambridge crystal database. And some of them have up to 5 copper atoms surrounding a single alkyne. So copper has some sort of strange affinity for the pi electrons of alkynes. You can also bind other metals to alkynes. Silver and gold are 2 of them, but they don't work as catalysts because they don't leave the product. So the silver ions are simply just attached, and the gold ions are simply just attached after the reaction. So you don't get a catalysis. You need 1 equivalent of gold, which is pretty expensive, right? Whereas copper has this exact affinity that is necessary for it to leave the product by proteolysis or by reaction with an electrophile. With the electrons bound to the copper, this can release the copper 1. So you have a transfer of the electron density to an electrophile from the copper atom that you don't see with the gold. So that's one reason that we don't use those. There are probably other atoms which could be used, like if you use ruthenium compounds, these will give you the 1,5 triazoles, not the 1,4 triazoles. But they are not like click reactions because you get, on a good day, you would get a 75% yield of your product, which is not what we are looking for. We are looking for quantitative chemical transformation. So, of course, there are other metals, but these are not efficient like the copper. And if you could just display the question again, because I think it was the second part.
Yes. Sure.
So the CuAAC ensures a perfect match because of the orbital geometry. The orbital geometry in the bis copper adduct of the alkyne. So the LUMO of that bis copper alkyne, which is the empty orbitals, fit exactly with the phases and the geometry of the azide. And that is why this is so efficient and why it's completely different from the Huisken reaction. The Huisken reaction is a true 3 plus 2 cycloaddition reaction, whereas this one is not a conventional cycloaddition. It gives a 3 atom partners react with each other to give a 6-membered ring.
Yeah, thank you, sir. So this is another question by Dr. Veena Nautiyal. It's also from Uttarakhand. Is chemistry solving the problem of motor neuron disease? If any solution, please let me know.
Yeah, so all diseases is chemistry gone wrong, essentially. And the only solution to that is chemistry done right. So that means that if you have a neuron deficiency of some kind, there is somewhere out there a chemical solution to that problem. And there's only a chemical solution to that problem. The chemical solution could be a very healthy diet and a lot of running, early up in the morning, early to bed at night. All of these things can actually make a huge difference, but the way they make the difference is by the chemistry that occur in the brain by that exercise.
Thank you, Professor. So there is 7th question by Sneha Saw, Ideal Engineering Institute of Engineering. She's asking, in your experience, what's the process of approaching unsolved problems, and how do you train yourself to ask the right questions that move the research forward? So this is.
So unsolved problem is always what you should work on in science, right? You want to work on unsolved problems and you want to come up with a solution for them. But the way to do that is to design an experiment that will reveal the details of the problem, reveal where is the factors that makes this unsolved, and then try to find the instrumentation and the chemistry that will allow you to ask that question and answer it by the analytical data that you get from that. So you will try, they say you want to find a new reaction. My favorite thing is to take a plate and make 100 reactions. And in 95 of them, nothing happens, but in 5 of them, something happened, and you don't know what it is, so you're looking for that. That's a very, very nice experiment.
Thank you, sir. So it's a question number 8 by Devdutt, Himachal Pradesh University, Simla. What is the scope of quantum computing to simulate the biomolecules or large molecules for drug application and analysis of complex structure of large molecules?
Well, so quantum computing and in particular quantum mechanics or quantum chemistry, in my opinion, we cannot calculate. So as I said before, we cannot calculate molecular recognition. We cannot calculate molecular recognition even using the quantum mechanics methods. We try, but we don't. It doesn't work. And I think there's probably something wrong with the Hamiltonians that we're using, in my opinion. But it's very, very difficult to show. I tried, and it doesn't really work. But in my opinion, we are not treating the antibonding orbitals as receiving electrons density, which they should. And that is maybe a factor that is missing in our quantum computations. So we are in force field calculation. We replace or we add hydrogen bonding to the force fields. So we actually, this is describing the donation of electron density from the lone pair of one part, the donor, or the acceptor of the hydrogen bond, into the donor of the hydrogen bond antibonding orbital, which leads to an elongation of the bond between the hydrogen and the heteroatom. And it leads to a short bond, a hydrogen bond between the lone pair and the hydrogen, so the antibonding into the antibonding orbital of the hydrogen. And I think that is actually not exclusive. Why should it be exclusive to the hydrogen? It's just easy to the hydrogen. But it should actually be part of every single bond that you have an antibonding orbital that should be involved. So this is what I'm trying to work on at the moment, to see if I can find a good experiment that will show that. Because this means that we have to rewrite quite a lot of math.
Thank you. So another question by Sautriki Pati, Techno India Group. So she's asking, sir, how did the concept of click chemistry evolve during your research, and what were the key scientific principles that guided its development? Might be somewhat repetitive, but yeah, sir, you can answer.
Yeah. So the click chemistry was originally by us carried out with a copper 1 iodide. And that was probably lucky because copper 1 iodide form large clusters where the clusters can actually have 2 copper atoms in the right distance for the click reaction. So the problem was, of course, the protection of the copper 1 state. If we were very careful with deoxygenation, we could go down to 0.001 0.0005 mole% of catalyst, which is quite low for a catalytic reaction. So it was extremely efficient. But we found that with just presence of small amount of oxygen, this didn't work. And that is, of course, one of the developments was how do we protect copper 1 in our reactions? How do we get the right source of soluble copper 1? And how do we protect it from the oxidation to copper state, oxidation state 2? And this has been, and also if we have macromolecules, the alkyne and the A side react very readily, but they need to be able to see each other. So if you have aggregates, you really need to deaggregate your molecules in order to get them to work. So this is another observation we did, that you probably have, in many occasions, you have an advantage of using urea or guanidinium chloride or something else to solubilize your alkyne or A side. Yeah, that's a very large number of small tricks that were invented along the way.
Thank you, sir. So
Also in the reaction, it's very important if you are cyclizing, using this for cyclization, that you have a very low concentration of your molecule, because if you have a larger concentration, it has a tendency to form oligomers instead of a cyclization. So we actually use an infusion pump in all our A-side alkyne reactions, where we take, for example, a protein. We need to stable the protein somewhere. So we take the protein in solution and we inject it very, very slowly into a click reaction mixture.
yeah, that is one of the issue with proteins, because they aggregate too quickly. And
yeah, when it's macromolecules tend to give you a lot of problems that you don't expect, which are not associated with click chemistry.
Thanks, sir. So yeah, the question is coming by Subhranshu Sekhar Samal from NIT Arunachal Pradesh.
Yes.
What are the challenges faced when we try to use nanoparticles in enzymes?
I'm not sure what that means. What are the challenges faced when we try to use nanoparticles in enzymes? You mean enzymes in nanoparticles? I don't know if we can communicate, because there's something.
Yeah.
There's something strange with the order of the words.
Maybe, sir.
Is it possible to talk with the person?
Not now, sir. It's because
okay,
too many people.
Then we have to, because I cannot see what is meant by this actually.
So maybe the order, maybe I think it could.
If I just take enzymes in nanoparticles, they can be plenty. Enzymes in nanoparticles probably at a high concentration of enzymes. If it's proteases, these will cleave each other. If it's nanoparticles made from peptides, they can also be cleaved by proteases. If it's other kinds of enzymes, could be that they are aggregating inside the nanoparticles due to the pressure concentration. So there are many, many different things that could be a problem. Okay. I don't know what enzymes we are talking about here.
Yes, we can go to next question. So this is very interesting question by Manik Bhomik from IC Bangalore. And he's asking that how it feels in Copenhagen. So many legendary Nobel laureates like Niels Bohr worked in that city in early 20th century. What is the environment there in the science community?
So yeah, that's a long answer. First of all, of course, it's a great honor to follow in the footsteps of both Niels and Aage Bohr. But we had actually here at Copenhagen University 14 Nobel laureates over time. Sorry, that must be in Denmark. And the last one is 25 years ago. So I'm the only one here now, and it's a great honor. If you look at the Copenhagen, so Denmark has a fantastic advantage because we pay very high tax. You wouldn't say that's not an advantage. You would say that's not an advantage, but it is an advantage now. It's a paying back that we have this high tax rate. In particular, 20, 30 years ago, we were the most taxed people in the world. In order to maintain a good society with a healthy structure. And what happened was that the industry was allowed to make foundations that own the company, and you could put money into the foundations without paying tax. So this was very lucrative for the companies initially. Eventually, the foundations grew much larger, and they actually started to invest in other companies. So they were not only the mother company, but a lot of different interests in the foundations. And they started to support more generally research in Denmark. And this is the reason that Denmark is per capita the richest country in Europe, and also one of the countries with most funding for research. So we are very lucky in that sense. And it stems back 200 years to the time where you had small villages in the countryside, and they were with farms sitting like that in a circle, and then you had all the fields going out like that. And it was like a real community, so they were sharing all their instrumentation and harvest machines and so on, and they were helping each other during the harvest. And this is sort of a sensation, an attitude that is engraved in Danes and is going through everything in the society. So building a community is, in my opinion, educating our children, and building a community like that is the most profitable thing you can ever do. Education, 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 that is where we generate most of our wealth. It's the best investment you can do. It takes time to get the return, but it's the best investment ever.
Really inspiring, sir. So, getting some other questions also. It is also related to artificial intelligence. So, it is Rajesh from IT Madras. He's asking, Click Chemistry has revolutionized the way we think about molecular synthesis. As the field progresses, how do you foresee the integration of click reactions with the new approaches, such as AI or automated synthesis platform to accelerate discovery and innovation in chemistry?
Yeah, so I've worked with automated synthesis all my life, and it's still just a tool. I can also see that I can handle it very well, but if you take a new students and they just have to press a button, so they don't really know what they're doing, it just doesn't work, because machines will always just be machines. There's nothing intelligent about artificial intelligence, which is a surprise to many people. It's just a machine. It's a new tool in the box that we should use as much as we can. We should try to avoid to use it for defense purposes, but we can use it, both machine learning and artificial intelligence, we can use it for helping us making decisions, and also finding much faster a way through a very large amount of data, to analyze the data, to come out with new solutions and so on. And the new solutions, you should remember, will always just be recombination of existing stuff.
Thank you, sir. So we are almost nearing to our conclusion, but still there are some few questions left. So it is from Rajesh Pandit, Aizer, Kolkata. What's the role of mathematics in understanding chemistry? In nature's working, does mathematics play a fundamental role? Or chemistry?
Yes, of course it does. I mean, quantum chemistry is mathematics, right? And so the problem with that is that the quantum, the mathematics people, or the quantum chemistry people mainly see the quantum chemistry as the truth and the reality to something that must adapt to the quantum chemistry. So this is sort of a sensation you get when you talk with people in quantum chemistry. And I think that's completely wrong. And we always thought that we knew everything, and we never did. So this is going to be an ongoing thing. We will never know everything. We'll never know the solution to all of these things that I've also alluded to today. But we will come closer and closer. And artificial intelligence, for example, is a really, really good help in trying to uncover the things that we really don't know today. What is it that we don't know? And mathematics plays a huge role because every time we find something new, we also have to make a mathematical description of it. And when we have that mathematical description, if it's accurate, it will help us to predict and foresee other things.
Thank you, sir. This question by Sthanu, it's a senior secondary school student. He's a school student. So he's asking that, what book would you recommend to get started on the basis of biochemistry for someone who just completed high school?
Okay, I'm not a biochemist. I'm an organic chemist. So I don't actually know all the biochemistry literature to get you a real good recommendation there. So what I will suggest to you is that you call a willingly professor at the university in the biochemistry department and ask them, because what I can recommend to you is only the chemistry book. I have a course which is on very complex stereochemistry, how you can actually synthesize all natural products, how you can make a design of a synthesis of a natural product, from understanding all the bonds in the natural product and how you can form them and so on. I think that's a very, very nice experience to teach the students this, because it involves a complete understanding of all the orbital theories and all of that. But biochemistry, DNA, proteins, and so what they do and so on, I am of a limited help there. I do know a lot about biochemistry, but it's not from a single book. It's from all sorts of picks and chooses from literature, reading a lot of articles and so on. So that's actually a recommendation. If you have a particular interest within biochemistry, just go on to Google Scholar. So Google Scholar, and then you just search for all of these things. Find all the ways you can combine words that will describe what you are interested in, and then you will find a lot of articles. You may actually take contact. If you are a very interested student, you might take contact to the university and make them help you get the actual documents, because some of them are, of course, with a fee. It's so irritating. You come into a web page and see the articles, but you cannot get it unless you pay like $50 or whatever. And I think that's not affordable. So line up with somebody who you think is interested in maybe looking at you as a possible subject for future studies, and see if you can get some of the articles that really describe what you're interested in.
Yeah, thank you, sir. So this is, I think, the last question, and it is very different type of question. How can chemistry contribute to demystifying boundaries of the multi-layered consciousness spectrum, unconsciousness, subconsciousness, consciousness? It is by Dev.
So the mind and the consciousness is an extremely complex concert of chemical reactions and chemical things that happens in the mind, in the brain. Nothing, not even our mind, is unbound. It's all anchored in whatever happens between the molecules in the brain. So there, the processing, the recognition, the signaling, and the transport, all of those things happen simultaneously in many, many molecular complexes all over the brain with all the memories we have and so on. And that's what creates the mirror of the universe, I call it, which is probably 3.5 billion years old in its evolution. So quite a complex thing, and all of it is chemistry, but it will take quite a while before we can use anything particular to describe this in a more holistic manner, to give us a picture of what the mind is doing. Conscious, unconscious, and so on. I don't know about unconscious because that's just not there, in my opinion.
Yeah. So coming to the conclusion of this question as a session, sir. So thank you
Yes.
very much, Professor Meldal, for your insightful response and addressing this question with such a depth and clarity. And also, most important, sharing your scientific journey with us